I'm going to quickly sketch out for you the heart. I'm going to draw as kind of a Valentine's Day heart just for simplicity, and also going to draw out some blood vessels. So let's draw the three big blood vessels coming off of the aorta. There are three major ones coming off the arch specifically. And then this first one, it branches off again. And so it's going to have a little branch there, something like this, and then it's going to go up. And this second one is going to go up as well. And I'm going to focus on these two that are kind of going up. The first one I'm labeling here is the left common carotid artery. Remember, these are all arteries because they're going away from the heart, right? So we know that these are arteries. And this one over here is the right common carotid artery. And these two arteries, the ones I've labeled, are going to kind of bulge and split. And I'm going to label the vessels that are splitting off. They're going to be one that comes to the inside, we'll call it internal, and one that goes further out to the outside, and we'll call it external. So this, for example, would be the external, and this one coming in would be the internal. And of course, this other one is also going inwards, and this one's going outwards. So it's labeled kind of the same way. So for example, if you want to name this, this would be the right external carotid artery, and this would be the right internal carotid artery, and the same for the other side. Now you remember we talked at one point about the carotid sinus. We had labeled a spot right here, and to be more accurate, maybe even slightly up into the internal carotid artery on both sides. These little X's then would be the carotid sinus. So let me just kind of write that out just to get us back and oriented to what we're talking about. And then of course there's this other spot I'm going to mark with an X down here. This would be the aortic arch, right? So I'm going to write aortic arch. Just to kind of remind us what's going on there, we had said that there are special nerves that are basically in the walls of the vessels there that can detect stretch. And those nerves, we'd call them baroreceptors. Remember, there are very famous baroreceptors, and those baroreceptors helped us regulate our blood pressure. So just want to refresh your memory about that. Now, what we're going to talk about is something slightly different. We're going to talk about chemoreceptors. Chemoreceptors. So chemoreceptors, they're not detecting stretch. These guys, they're actually detecting some chemical, and that chemical is going to be either oxygen or carbon dioxide. And whenever I mention carbon dioxide, I also want you to remember protons, and we'll get to why those are connected as well. So where are these special chemoreceptors? Are they in the sinuses, in the arch? Well, yes and no. They're close by, so they're actually, for example, you have these carotid bodies of tissue right here, close by, but not exactly the same spot. They're not in the walls, they're actually little bits of tissue, right? And we call them little bodies of tissue that are close by to where those baroreceptors are, but they are distinct. They're different locations, and they certainly have a different function. And because they are called bodies of tissue, we actually call them that. We call them carotid bodies, and you might even hear the term aortic body. Aortic body would be the one that I drew on that aortic arch. So these are the chemoreceptors. I'm just drawing a little arrow to point them out, make it very clear where these things are located. And you can see that they're close to those green X's, but not necessarily the same thing. Again, the X's are actually in the walls, and the carotid bodies and the aortic bodies are actually bodies of tissue. Maybe I'll write that out. Bodies of tissue. So you can actually imagine a little nubbin of tissue being these specific carotid bodies or aortic body. So if this is what it looks like, let's blow it up and get a better view of it. I'm going to expand this white box and this white box. I'm just going to make them bigger. So the first one is basically going to look like this, right? You've got a little junction of two vessels, and off the external carotid artery, you might have little branches, maybe little arterial branches that break up into tiny little capillaries. And if you looked really closely, you'd see a lot of little blue cells here. Blue cells, each getting fed by a different capillary, and that's how they're getting their nutrition, right? And if you actually looked at all these blue cells kind of in aggregate, you would see that you could consider this as a body of tissue. And the same thing over here with the aortic arch. I've got my aortic arch right here, but if I actually look closely, there'd be little branches coming off of the aorta and supplying this area with much needed oxygen, right? So little branches off this aorta breaking up into tiny little capillaries, and those capillaries are actually serving same thing, a little body of tissue. And this is all the little cells that make up the aortic body. So now you get a sense for when I say the aortic body and carotid body, it actually is a little body of tissue made up of little, what I've drawn here is blue cells. And one reason I wanted to actually draw it out this way is that the carotid body in particular, this guy over here is getting lots and lots of blood perfusion. Remember we talked about the idea of perfusion as the flow to a certain amount of tissue. It turns out that the carotid body gets about as much as anywhere in the body. It gets about 2 liters per minute for every 100 grams of tissue. Now think about that. If you have, for example, a 2-liter bottle of soda, just to give you perspective, let's say you have a little 2-liter bottle of orange soda or something. If you were to pour that 2-liter bottle out on top of something that's 100 grams, maybe like a tomato, let's say this is a tomato that's 100 grams, if you could do that in 1 minute, then that would be the amount of perfusion that your carotid body is getting. That's a lot of flow of blood that's going through those capillaries to the carotid body. And the aortic body also gets well perfused, but it's pretty striking. This number is pretty amazing when you consider this in terms of a real life experiment, like putting soda on a tomato. So I'm going to blow this up one more time, and what I want to just have you remember is that lots and lots of blood, I just said, is gushing through these capillaries, serving these cells. And these cells are actually given a very funny, interesting name. I'm going to try to draw it as a circle to help you remember that these are globus cells. So it almost looks like a globe. And these globus cells, what they basically do is they're actually going to detect what's going on in the blood. So in the blood you've got a little red blood cell, and this little guy has got some hemoglobin, and you have a little molecule of oxygen. This is oxygen. And some of the molecules are going to be bound to the hemoglobin, and some are going to be kind of just floating, dissolved in the blood. And over time, that oxygen might kind of diffuse into this globus cell, something like that. So over time, the globus cell is kind of just sensing what's going on in the blood in terms of oxygen levels. And if levels start to fall, it's going to know it because less oxygen is diffusing inside. It's going to feel that there's less oxygen inside the cell. And if that were to happen, this cell depolarizes. The membrane depolarizes. And so it's going to send a signal down here. It's going to depolarize. And if it does depolarize, what happens on the other end is that you actually have little vesicles here, and they're full of some sort of neurotransmitter. So if these vesicles sense that there's a depolarization going through, they're going to dump all that neurotransmitter out right here. And on the other side, you actually have a nerve. So nerve is going to sense that, and it's going to say, hey, I've got some neurotransmitter here. Let me fire off an action potential. Now, if you step back and look at this, you're noticing certain things about the globus cell. You're saying, well, wait a second, this globus cell, it is able to depolarize, and it actually has little vesicles full of neurotransmitter. So what does this sound like? It sounds, although I haven't drawn it this way, it sounds like it's kind of like a nerve, right? It has depolarization and neurotransmitter. It turns out that it's actually made of this, or it comes from this material called neuroectoderm, which in other words, these globus cells I'm going to draw here, they're basically derived from the same types of cells as gave rise to our nervous system. So our nervous system and these globus cells both have kind of a shared common ancestor. So if you go back to the developing baby, you'll find a common ancestor, and we call that ancestral type tissue neuroectoderm. So it's just kind of interesting to note that these globus cells are very, very closely related to nerves, although we don't think of them as nerves themselves. So far so good. I said that you have, a trigger, which is oxygen, for this depolarization, and specifically it's going to start depolarizing a lot more if the level of oxygen starts to fall. So if this globus cell says, well, wait a second, I'm noticing a low level of oxygen, low partial pressure of oxygen in the blood, it will start sending more signals. It's going to start firing off more and more of these signals down here because it's going to dump more neurotransmitter into that nerve. So this becomes a major signal, low amount of oxygen. Now another signal is carbon dioxide. So of course, This globus cell is like any cell. It's making carbon dioxide, and over time that's going to kind of start diffusing into the blood. But let's say that the blood is loaded with carbon dioxide. Let's say it's just full of it. Well, if it's full of carbon dioxide, then there's not going to be much diffusion into the blood, right? Because there's no gradient anymore, right? So this carbon dioxide is going to sit around. It's going to sit inside the cell. And this becomes another trigger. So high amounts of carbon dioxide become another trigger for depolarizing the globus cell. And again, of course, if it depolarizes, you're going to send more action potentials down this way, and more action potentials from that nerve tell the brain eventually that, hey, there's a lot of carbon dioxide or there's a low amount of oxygen, one or the other. So a second signal then is carbon dioxide. Now remember this formula. Carbon dioxide is also going to enjoy binding with water. Remember, it always enjoys that relationship, and so it forms carbonic acid, and that splits up into bicarb, right? This is something that we know happens all the time. And what you get then is this relationship. Anytime I mention lots of CO2, in your head there should be little bells going off saying, then he also must mean lots of protons, because if you have a lot of CO2, you also have high protons. And we know that high protons, another way of saying that is low pH, a low pH. So just remember, the triggers then for the globus cell are a few, right? You could have a low amount of oxygen, a high amount of carbon dioxide, or a high amount of protons, in other words, a low pH. So these are the triggers to make that nerve fire. So shifting back to this picture, if these cells are triggering nerves, then these nerves are going to kind of combine up, and both in the aortic body and also the carotid body, they're going to kind of form one giant nerve. And you literally have, at the end of the day, a giant nerve kind of going back from these two carotid bodies and also from this aortic body. The ones coming from the carotid bodies are called the glossopharyngeal nerve. The glossopharyngeal nerve. This is just kind of a long name, but you'll definitely come across it at some point, so I wanted you to be familiar with it. And the nerve coming off of this aortic body is the vagus nerve. Vagus nerve. A little bit like Las Vegas, but probably not as fun, but more useful. So I haven't said it explicitly, but these chemoreceptors then are your peripheral chemoreceptors. Peripheral chemoreceptors. And now you know how these things work and the nerves connected to them. So I hope you enjoyed that.